In this video, I will be explaining the BSc 5th semester subject Zoology, Immunology and Animal Biotechnology as prescribed by Osmania University and other universities of Telangana. We will go through the important questions and answers one by one. Let's begin with a quick revision. Unit 4 first important short question is 1. In vitro fertilization, IVF. In vitro fertilization, IVF, is a technique in which fertilization happens outside the body in a laboratory. The word in vitro means in glass, so the egg and sperm are allowed to fuse in a glass dish instead of the female reproductive tract. How IVF works? Simple steps. 1. The woman is given medicines to produce multiple eggs. 2. Mature eggs are collected from the ovaries using a fine needle. 3. Sperm is collected from the male partner. 4. Eggs and sperm are mixed in a petri dish, where fertilization occurs. 5. The fertilized egg becomes an embryo. 6. A healthy embryo is then transferred into the woman's uterus, where pregnancy begins. Example: if a couple cannot conceive naturally due to blocked fallopian tubes or low sperm count, IVF helps by bringing the egg and sperm together in the lab. In simple words, IVF is equal to baby formation outside the body plus embryo placed back inside the uterus. It is one of the most successful assisted reproductive technologies used today. Next important short question is 2. Embryo transfer. Embryo transfer, ET, is a biotechnology method in which a healthy embryo is placed into the uterus of a female so that it can grow into a normal baby. This technique is commonly used in IVF programs, animal breeding, and livestock improvement. How embryo transfer works? Simple steps. 1. First, eggs and sperm are allowed to fertilize outside the body, as in IVF, or inside the donor female. 2. After fertilization, the embryo grows for a few days, usually till 4 to 8 cell stage or blastocyst stage. 3. A healthy embryo is carefully selected using a microscope. 4. It is loaded into a thin, flexible tube called a catheter. 5. The embryo is gently placed into the uterus of another female, called the surrogate mother or recipient female. Example: in cattle breeding, embryos from a high-quality cow are transferred into ordinary cows. This helps produce genetically superior calves more quickly. In simple words, embryo transfer is equal to taking a good quality embryo and placing it in a female's uterus for pregnancy. This method increases success rate in both humans and animals. Next important short question is 3. Hybridoma technology. Hybridoma technology is a technique used to produce monoclonal antibodies, special, identical antibodies made from a single type of immune cell. These antibodies are very important in medicine, diagnostics, and research. How hybridoma technology works? Simple explanation. 1. A mouse is injected with an antigen, foreign substance. 2. The mouse's body produces B lymphocytes that make antibodies. 3. These B cells are taken out and fused with myeloma, cancer cells. 4. The fused cell is called a hybridoma. It has two powers. B cells make antibodies. Myeloma cells divide endlessly. 5. Hybridoma cells are grown in culture to produce large quantities of a single type of antibody. Example: monoclonal antibodies produced by hybridoma technology are used in pregnancy test kits, cancer treatment, COVID-19 test kits, diagnosis of infections. In simple words, hybridoma technology is equal to joining an antibody-producing cell with a cancer cell to make unlimited antibodies. It is one of the most powerful tools in biotechnology. Next important short question is 4. Stem cells. Stem cells are special cells in our body that have the amazing ability to divide and change into different types of cells. They act like the body's repair system because they can replace damaged or dead cells. Key features of stem cells. 1. Self-renewal. They can keep dividing for a long time. 2. Differentiation. They can change into specific cells like nerve cells, muscle cells, blood cells, etc. Types of stem cells. Simple. Embryonic stem cells, found in early embryos, can change into any type of cell in the body. Adult stem cells, found in tissues like bone marrow, can form limited types of cells. Induced pluripotent stem cells, iPSCs, normal body cells changed back into stem cells using biotechnology. Example. Bone marrow stem cells are used to treat blood cancers like leukemia. Doctors replace the patient's damaged blood cells with healthy stem cells. In simple words, stem cells is equal to master cells that can make new cells and repair the body. They are very important in medicine, cloning, and regenerative therapies. Next important short question is 5. Bacillus thuringiensis. Bacillus thuringiensis, BT, is a naturally occurring soil bacterium that produces a special protein toxin harmful to insects but safe for humans, animals, and most plants. Because of this, BT is widely used as a biopesticide in agriculture. How BT works? Simple explanation. 1. BT bacteria make crystal proteins called cryotoxins. 2. When insect larvae, like caterpillars, eat BT, the toxin reaches their alkaline gut. 3. In the insect gut, the toxin becomes active and creates holes in the gut lining. 4. The insect stops eating, becomes weak, and eventually dies. 5. BT affects only specific insects and does not harm beneficial insects like honeybees. Example. BT cotton is a famous genetically modified crop. It contains the BT gene, so the plant itself produces the toxin and protects itself from bollworms without chemical pesticides. In simple words, Bacillus thuringiensis is equal to friendly bacteria that kills harmful insects naturally. It is an eco-friendly biopesticide used to protect crops and reduce chemical pesticide use. Next unit 4. Long questions. First important long question is 1. Describe hybridoma technology concepts and applications. Hybridoma technology is an important biotechnology method used to produce monoclonal antibodies, which are identical, highly specific antibodies made from a single type of cell. These antibodies are widely used in medicine, diagnostics, and research. Concept of hybridoma technology, simple explanation. Our body has B lymphocytes that make antibodies, but they cannot divide forever. Cancer cells, myeloma cells, can divide endlessly, but they do not produce antibodies. Hybridoma technology combines the best features of both. Steps in hybridoma technology. 1. Immunization. A mouse is injected with an antigen, foreign substance. Its B cells produce antibodies against that antigen. 2. Isolation of B cells. Antibody-producing B cells are collected from the mouse's spleen. 3. Fusion with myeloma cells. B cells are fused with immortal myeloma cells using PEG, polyethylene glycol. This fusion creates hybrid cells called hybridomas. 4. Selection of hybridomas. Only fused cells survive in HAT medium. Unfused cells die, ensuring pure hybridoma culture. 5. Screening. Hybridomas that produce the desired antibody are identified and selected. 6. Large-scale production. Selected hybridomas are grown in culture to produce monoclonal antibodies, MAbs, in large amounts. Applications of hybridoma technology. 1. Medical diagnosis. Pregnancy test kits. Detection of viral infections, HIV, hepatitis, COVID-19. Cancer biomarkers. 2. Disease treatment. Monoclonal antibodies used in cancer therapy, example, rituximab, trastuzumab. Autoimmune disease treatment. Antivenom production. 3. Research. Identifying specific proteins. Studying cell behavior. Drug testing. 4. Biotechnology. Producing highly pure antibodies for experiments. Quality control in industries. Simple example. In a COVID-19 rapid test, monoclonal antibodies bind to viral proteins. This produces a visible line on the strip. These antibodies are produced using hybridoma technology. In simple words, hybridoma technology is equal to joining a B cell, makes antibodies, plus cancer cell, divides endlessly, to produce unlimited monoclonal antibodies. It is one of the most useful tools in modern biotechnology and medicine. Next important long question is: 2. What are stem cells? Write about its types and applications. Stem cells are special cells in the body that can divide continuously and can change into many different types of cells. Because of these abilities, stem cells act like the body's repair and renewal system. They replace damaged or dead cells and help in growth and healing. Types of stem cells. 1. Embryonic stem cells. Found in early-stage embryos. Can form any cell type in the body, pluripotent. Very powerful, but their use raises ethical concerns. 2. Adult, somatic, stem cells. Found in tissues like bone marrow, brain, skin, liver, etc. Can form only a limited number of cell types. Important for natural repair. Example, bone marrow stem cells make all blood cells. 3. Induced pluripotent stem cells, iPSCs. Normal adult cells, like skin cells, turned back into stem-like cells using biotechnology. They behave like embryonic stem cells, but avoid ethical issues. 4. Cord blood stem cells. Found in the umbilical cord of newborns. Useful in treating blood-related disorders. Applications of stem cells. 1. Regenerative medicine. Repairing damaged tissues like heart muscles, spinal cord, or cartilage. Used in treating burns and wound healing. 2. Treatment of diseases. Used in bone marrow transplant to treat leukemia and lymphoma. Potential to treat Parkinson's, diabetes, and Alzheimer's in future. 3. Drug testing. New medicines can be tested on stem cell-derived tissues instead of animals. 4. Research. Helps scientists understand how diseases develop. Useful in genetic studies and developmental biology. Example, easy to remember. If a patient has damaged heart tissue after a heart attack, doctors can use stem cells to grow new healthy heart cells. This helps the heart recover faster. In simple words, stem cells is equal to master cells that can make new cells and repair the body. They have huge importance in medicine, research, and biotechnology, and are considered the future of advanced therapies. Next important long question is: 3. What are animal bioreactors? Write a brief note on its concepts and application. Animal bioreactors are specially designed living animals that are genetically modified to produce useful biological products, such as medicines, hormones, or proteins. These animals act like biological factories, making large quantities of valuable substances in their milk, blood, eggs, or urine. This process is a major part of animal biotechnology and helps in producing important products at a low cost. Concept of animal bioreactors. Simple explanation. A bioreactor normally means a machine where cells grow and produce products. But in animal bioreactors, the animal's body acts as the bioreactor. Steps easy to understand. 1. A helpful human gene, example, insulin gene, is inserted into an animal's DNA. 2. This gene is targeted to express in a specific part, usually the mammary gland, milk-producing tissue. 3. When the animal produces milk, it also produces the desired protein in the milk. 4. Scientists collect the milk and purify the product. Animals commonly used: goats, sheep, cows, rabbits, chickens produce protein in eggs. Applications of animal bioreactors. 1. Production of medicines. Human insulin, human growth hormone, blood clotting factors for hemophilia, antibodies used in cancer and COVID-19 treatments. Example: ATRYN, an anti-clotting protein, is produced in the milk of genetically modified goats. 2. Production of vaccines. Animals can be engineered to produce viral proteins, which are used to make vaccines. 3. Organ and tissue production. Research is going on to grow organs for transplantation inside animals like pigs. 4. Industrial enzyme production. Animals can produce enzymes used in food, detergent, and pharmaceutical industries. 5. Agricultural benefits. Genetically modified animals can grow faster or become resistant to diseases. Simple example to remember. Imagine a goat producing life-saving medicine in its milk. This goat becomes a bioreactor, just like a factory producing useful products. In simple words, animal bioreactors is equal to genetically modified animals that produce valuable medicines or proteins in their milk, eggs, or blood. They are low-cost, efficient, and extremely helpful for modern biotechnology and medicine. Next important long question is: 4. Explain concept of biopesticides. Biopesticides are natural pesticides derived from living organisms like bacteria, fungi, viruses, or plants. Unlike chemical pesticides, they are eco-friendly, biodegradable, and target-specific, meaning they kill pests without harming humans, animals, or beneficial insects. Biopesticides are an important part of sustainable agriculture. Concept of biopesticides. Simple explanation. Pests damage crops and reduce yield. Traditionally, chemical pesticides were used, but they have many problems. Pollute soil and water. Harm non-target organisms like bees and fish. Cause pesticide resistance. Biopesticides solve these problems because they are derived from natural sources and act specifically on pests. They work by interfering with the pest's life cycle, feeding, or reproduction. Types of biopesticides. 1. Microbial biopesticides. Made from microorganisms like bacteria, fungi, or viruses. Example: Bacillus thuringiensis, BT, produces toxins that kill caterpillars. 2. Plant-incorporated protectants, PIPs. Genes from natural pesticides are inserted into crops to make them resistant to pests. Example: BT cotton contains the BT gene and kills bollworms. 3. Biochemical biopesticides. Naturally occurring chemicals that control pests. Example: neem extract repels insects and prevents growth. Mode of action, example, Bt toxin. 1. Caterpillars eat Bt-treated leaves. 2. Bt toxin reaches the insect gut and breaks gut cells, stopping digestion. 3. The insect stops eating and dies. This action is specific to insects and safe for humans, birds, and beneficial insects. Advantages of biopesticides. Environmentally safe and biodegradable. Target specific, reducing collateral damage. Do not leave harmful chemical residues. Reduce chances of pest resistance. In simple words, biopesticides is equal to nature's own pest control. They protect crops using bacteria, viruses, fungi, or plant compounds instead of harmful chemicals, keeping agriculture safe and eco-friendly. Example to remember. Bt cotton and neem extract sprays are common examples used by Indian farmers to fight pests naturally. They are smart, green, and safe alternatives to chemical pesticides. Next important long question is: 5. Write about the procedure involved in the transfer of an embryo during in vitro fertilization. Embryo transfer in vitro fertilization, IVF, is a critical step in assisted reproductive technology, where a fertilized embryo is placed into the uterus of a woman so that it can develop into a healthy baby. This procedure helps couples who are unable to conceive naturally due to fertility problems. Procedure of embryo transfer, simple explanation. 1. Ovulation induction. The woman is given hormonal medicines to stimulate the ovaries to produce multiple eggs instead of the usual one. This increases the chances of successful fertilization. 2. Egg retrieval. Mature eggs are collected from the ovaries using a thin needle guided by ultrasound. This is a painless, minor procedure done under local anesthesia. 3. Fertilization in the laboratory. The retrieved eggs are mixed with sperm from the partner or donor in a petri dish. Fertilization occurs outside the body, forming embryos. 4. Embryo culture. Fertilized eggs, embryos, are grown for 3 to 5 days in a special culture medium. Healthy embryos are selected for transfer. 5. Embryo transfer. The chosen embryo is loaded into a soft catheter. A doctor inserts the catheter through the cervix into the uterus carefully. The embryo is gently released into the uterine cavity. 6. Post-transfer care. The woman may be given hormonal support to help the embryo attach to the uterus, implantation. A pregnancy test is done after about 2 weeks to confirm success. Example to remember. If a couple cannot conceive due to blocked fallopian tubes, IVF plus embryo transfer allows fertilization in the lab and implantation in the uterus, bypassing natural barriers. In simple words, embryo transfer is equal to putting a lab-made fertilized egg into the uterus to grow into a baby. Think of it as planting a seed in the soil. Lab fertilization is equal to seed creation. Uterus is equal to soil. Hormonal support is equal to water and sunlight. This procedure is safe, effective, and widely used in humans and even in animal breeding programs to produce genetically superior offspring.